morning, everyone. Welcome back to the final day of our Rare Drug Development Symposium, hosted in partnership with Global Genes and the University of Pennsylvania's Perelman Medical School Orphan Disease Center. It is my distinct pleasure to welcome you back all today, and I'm so excited for some great sessions and discussions coming up. Now, before we jump into all of that, I just wanted to get some housekeeping remarks out of the way so that you can hear from all of our great speakers. And so I will be turning it over to Dr. Jim Wilson for his opening remarks in just a moment, but first wanted to review a couple of key details. First, the chat box to the left of your screen allows you to interact with other attendees, the organizers, and discuss the content of the day. We hope that you use this opportunity to make new connections. However, the chat does not allow for links or other documents, so if there are resources that you'd like to share, please be sure that you visit the social wall and post those there. We encourage you to share any and all resources with your peers, as we know that we cannot cover all of the information that is needed in the space in the 3 days that we have here, so please use this as an opportunity to share and be collaborative. Additionally, if you want to connect with any individual attendees, please use the attendee list function or the join the conversation chat on the side. The questions box is located to the left of your screen as well with the chat piece. Please do be sure that you are putting the questions into the questions box and not the chat. The speakers do not see the chat piece, so the questions are going to get to them much more easily, and the questions allow you to upvote each other's as well if there are key topics of interest. We really encourage you to utilize this time to ask your questions of the presenters and of each other as well. This is your time to find answers and insights to the questions and challenges that you're facing in your own work, and we want to ensure that you are finding answers and new opportunities to advance your partnership in research. And finally, to the right of your video, you will see captions and translations. Please, if you're looking to utilize either of those, just click in and select the language of your choice. And thank you to both Horizon and Greenwich Biosciences for their support of these accessibility tools in particular. Additionally, you can utilize the notes function to take notes in the player, and those will be emailed to you automatically after you close the session. If you have any technical difficulties within the session, please click on Request Support. If you are outside of a session and need additional support, please utilize the live chat button. We have a great team behind the scenes who are ready and able to assist you. And finally, we just want to remind everyone to please participate as active listeners and participants in this conversation. We know in a virtual and digital world, it is very easy to get distracted, and all of us have spent probably more time than we'd like to admit in Zoom rooms this year. While we're really looking forward to opportunities to be back together in person in the future, we hope that you'll all take the time to be present with us here today. While remote, we are thinking of all of you and are really grateful for an opportunity to be here in community with you. We appreciate you all tuning in, and without any further ado, I am so pleased to turn it over to my colleague, Jim Wilson, who is the Rose H. Weiss Professor and Director of the Orphan Disease Center, and also a Professor of Medicine and Pediatrics and Director of the Gene Therapy Program at the University of Pennsylvania, among probably quite a few other accolades and hats he holds. And so without any further ado, I will turn it over to Jim to share some opening insights. Thank you so much.
Thank you, Anne, for the introduction. And again, very much appreciate the partnership we have with Global Genes and the Orphan Disease Center. And I look forward to many other opportunities to collaborate. I am the Director of the Orphan Disease Center and the Director of the Gene Therapy Program. Just a couple thoughts about where we are. It's a very interesting time as we begin to enjoy a world with COVID in our rear view mirror. It is important to take stock of where we are with respect to rare disease research and where this research is heading. A tragic but underappreciated consequence of COVID is the negative impact it has had on the progress of non-COVID research, especially rare disease research. As we all know, labs were shut down and clinical trials were delayed, and precious time was lost, which is the one commodity those living with rare diseases do not have. But what should we expect as COVID restrictions are lifted? The good news is labs have repopulated and scientists are more energized than they were. The worst thing you can do to a scientist is tell them they can't do science. And clinical trials are reopening. And the therapeutic platforms that were available to us before the pandemic are still there and ready to be deployed for treating rare diseases. And they seem to grow with time, including gene therapy, genome editing, cell transplantation, and antisense oligonucleotides. In fact, it is rare diseases where many of these platforms have the greatest chance of success. And importantly, when we do achieve that success, it can be leveraged across many other like diseases, simplifying the drug development process. As a physician scientist who's been driving gene therapies forward for over 35 years, this day has been a long time coming. With that said, there is no room for complacency. The business challenges of one and done treatments for rare diseases are huge, and they significantly diminish the commitment of resources by traditional drug developers. We may be in the ironic position to be able to treat or potentially cure a number of rare diseases, but will be unable to do so because of lack of insufficient resources or lack of access to necessary infrastructure. As a cautionary note, however, please realize that research funding, while necessary, is not sufficient. There must be adequate scientific rationale to proceed into clinical testing. Just because a scientist says they can do something for you does not mean you should support them. But what can you do? And I guess that's why we're here. You need to drive the research agenda, particularly if you're being ignored by commercial and government funders. The bar for biopharm to take on your disease is much lower if you progress the science. And if they don't take on your programs, we will find nonprofit ways to do so. Last year, we created a nonprofit organization called the Institute for Life-Changing Medicines, whose remit is to develop and distribute transformative treatments for diseases that are tractable, but neglected because they're too rare. And I suggest there will be many other like organizations as the opportunity arises. Second, you need to use your pulpit as patient advocates to assemble partnerships. The drug development process is much too complex to try to go alone. And finally, you need to absolutely insist that critical data, such as natural history studies or toxicities that are observed in animal and clinical trials, are openly shared amongst all stakeholders, including the drug developers. This practice not only accelerates progress, but it will save lives. Anything short of full transparency in these critical issues should be considered immoral and should not be tolerated. Looking into the crystal ball of the rest of 21 and 22, I'm incredibly optimistic. I see positive clinical data emerging for many rare diseases using current platforms such as antisense oligonucleotides and cell and gene therapy. I also see the introduction into the clinic of many more applications of genome editing. So as final words, it is clear that the therapeutic revolution for rare has begun, and we're delighted that we can partner with you and all of those attending in this meeting to make sure that it realizes its potential. So now I'd like to welcome back Barbara Goodman, President and CEO of Cures Within Reach, to announce the winner of the Cure Accelerator Live for Rare Diseases. Thank you very much.
Good morning, everyone. I'm Barbara Goodman, and I'm joined this morning by Claire Thibodeau. We are here to announce the winner of the Cure Accelerator Live for Pediatric Rare Disease event, the competition that we held yesterday evening. Before we get to the winner announcement, just a quick information about Cures Within Reach. We are leveraging the speed, safety, and cost effectiveness of medical repurposing research, whether it has philanthropic purposes or commercial uses. As the COVID pandemic has shown to everyone, the fastest way to therapeutic impact and treating patients with unmet medical needs like COVID-19 is through the testing of already approved therapies in randomized controlled trials. As Jim alluded to just a few minutes ago, no one understands this better than the rare diseases community, who has been living in crisis mode from the moment of diagnosis of a rare disease. And we're thrilled that over a decade, we have been funding proof of concept, pivotal studies, first in human studies, to get the data needed for follow-on funding. In fact, our nearly 100 projects that we have funded have invested $6 million in researchers, in clinicians and PIs. And our researchers have follow-on funded. They've raised additional funds for the secondary clinical trial, the phase 2 trial, the larger confirmatory study of $67 million, a leverage and catalytic rate of nearly $10 for every dollar we've invested. Our organization is disease agnostic, we're geography agnostic, we're institution agnostic, and of course we do so much in rare diseases. Over 42% of our ongoing projects today are in the rare disease community. We're here today to select another project in the pediatric space, one of our major 2021 initiatives. Our other initiatives this year are making an impact in veterans, in PTSD and in depression. We're selecting those projects later this summer. And there's an open call for proposals right now. In fact, 3 RFPs in the minority and underserved community, both underserved researchers as well as minority patients and health disparities and in the developing world. But you're not here to hear about us. You're here to hear who won the Pediatric Rare Disease competition. I want to thank the Charles H. Hood Foundation for their partnership with us. We have been partnering with them for under a year. This is our first initiative with them. The Charles H. Hood Foundation was founded in 1942 and is focused on impacting the health and quality of life of children since then. They've been phenomenal partners, very collaborative, and very engaged with the selection of the winner today. I also, of course, want to acknowledge our other industry partners, our philanthropic partners, and other organizations that are so integral to Cures Within Reach and our mission. With that, I'm going to turn it over to Claire, who's going to talk about our finalists.
Thank you, Barbara, and thank you to everybody who attended the event last night and voted. Everybody who attended knows we had 4 fantastic finalists, all of whom had really impactful clinical trials to help rare disease patients and their caregivers. Doctors Gordon, Rauer, Tang, and Fuentes really did an outstanding job. However, this is the part where it comes down to who won the competition. And, you know, at this point, I usually say there is only one winner. But 2021 is a new year, and we are excited and thrilled to announce that we were able to select 2 winners this year. We are just so excited that we will now be supporting the clinical trials presented by Dr. Joyce Tang at Stanford University and Dr. Nancy Gordon from the MD Anderson Cancer Center. So I actually have Dr. Gordon on the phone with us, and I know she'd love to thank everybody who voted for her. Dr. Gordon, congratulations.
Thank you. I just wanted to quickly say a few words. I'm so really grateful for the Cure Accelerator, Cure Within Reach, and Partner Foundation's generosity for their provided support to our patients, who I'm sure will benefit from our investigations. I feel really honored to be one of the recipients. I really look forward to report back to you the results of the study. Thank you very much again.
Thank you, Nancy. We really appreciate you joining us this morning. So again, thanks to everybody who attended, everybody who voted. Your vote does count. And please know that we are very supportive of all the projects that, that presented, and we will continue to fundraise in the hopes of funding all of these fantastic trials. So now I'll turn it back over to Barbara for her closing remarks.
Wonderful. Thank you. And again, congratulations to both Drs. Tang and Dr. Gordon. We're thrilled with that. And as Claire said, we are actively fundraising for the other two finalists. Now we're excited to turn it back over to the keynote and the session, Seizing the Moment, Expanding Impact and Access in Translational Science. I'm excited to introduce the moderator for this session, Dr. Donna Cryer, the President and CEO of the Global Liver Institute.
Hello, my name is Donna Cryer. Barbara, thank you so much for such exciting news to start off this day of the Rare Drug Development Symposium. I am, as a rare liver disease patient, I am so honored to be part of this event, let alone to be moderating this keynote session on seizing the moment, expanding impact and access in translational science. Because we will be having a lovely conversation and Q&A session after Dr. Redder makes her remarks, I will remind you again to use the question portion on the left side of your box, as you were instructed earlier, so we can get your questions answered. I know there's so many. This is a wise, insightful, experienced group, and I look forward to seeing your questions as well as having many of my own. Dr. Joni Redder is the acting director at the National Center for Advancing Translational Science, or NCATS, as we all know it. Dr. Redder is responsible for planning and executing the center's complex and multifaceted preclinical and clinical programs. And before joining NCATS, she served us as the director of scientific programs within the All of Us Research Program, which is part of the Precision Medicine Initiative at NIH. I could go all day simply reading Dr. Redder's bio, but I think we're all here to hear her. So without further ado, Dr. Joni Redder.
Donna, thank you so much. It's a pleasure to be here, and hello everyone, and thank you for this kind introduction, Donna. And I'd like to also thank the organizers at Global Genes for the invitation to speak today. I can tell you that Dr. Wilson and I did not compare notes this morning before this session, but I will be reiterating some of the important points that he made in his opening remarks. And so I'm really excited to have also a discussion at the end with Donna, and I look forward to some of the questions that you might have as well. So I'm going to talk today about a variety of things, but really starting out first to frame the problem that NCATS is tackling, and to tell you a little bit more about translational science and how we think that this is a wonderful approach to help expanding the impact of access to the critical care that rare disease communities need. So seizing the moment is a wonderful title for today. So let's frame that problem. In a way, this is actually from Our World in Data, which is a website that you can go to. And if you look over the last 30 years of disease burden, this is measured in disability adjusted life years, or this is known as dailies. And essentially what that means is it's calculating the number of years of life lost due to premature death or disability from the burden of disease. And so if you follow this over time, you can see that it hasn't really changed much. Those disease clusters at the top haven't really changed much. But what they have done is gone in the wrong direction. We have more tools and more technologies, but we're still trending in the wrong direction. So in addition to that, we think that there are about 10,000 diseases total, probably more than that, but as a rough estimate. And most of these diseases are rare. And as you've probably heard these estimates before, but about 7,000 rare diseases affecting about 1 in 10 Americans, and that's some 30 million people. So for a long time, and what's been my life's work as a geneticist, we've been trying to figure out what genes cause what diseases. And there has been tremendous success in that work. But what's been much slower is translating that knowledge that a gene causes a disease into finding a treatment for that disease. In fact, only about 500 of all diseases have a treatment. So that's 95% of diseases then that don't. So for these genetic diseases that we know about, knowing the gene is among the first of many steps that are needed to consider how to develop these treatments. And then thinking about doing that on the scale of all diseases is daunting, to say the least. So we need to do something a little bit different. So our mission at NCATS is to turn promising research discoveries into health solutions. And our driving hope is to get more treatments to more people more quickly. The NCATS approach represents sort of each stage across the research continuum. And it's not very linear, or it's not really unidirectional, but each phase builds upon itself and informs the others. And at all stages of this spectrum, NCATS really develops new approaches and demonstrates their usefulness, and then disseminates its findings. And inclusive of this is the need to really hear from people who are affected by the diseases that need these therapeutic solutions. So importantly, inequity is a structural issue that has to be addressed. People with rare diseases from underserved communities have extra challenges. This is an inequity. We cannot stop at developing therapies. We must continue to work to raise awareness around the inequities in rare and common diseases within communities of color. And all that means, talking about awareness, to workforce development, to engagement, to access to care, to training the next generation. So again, our mantra has been to bring more treatments to more people more quickly, but I'm going to make a little edit here. We want to bring treatments to all people more quickly. So that is really our goal at NCATS. And essentially, the way that we do this is really twofold. Every project that we take on is about translation. That is, it has to advance the science of the project. So for example, moving a project from preclinical development, shown here in the blue chevron diagram, to the phase 1 clinical trials here shown in sort of this salmon pink color. And in addition to that, the second thing it does is we want to identify and address the costly and time-consuming bottlenecks that slow down or prevent the translation of this research. And so if you think about a pipe that flows from sort of A to B on this diagram here, things go in and things come out, and the flow is determined by the openness of that pipe. And pipes have bottlenecks. And so these are the things that I'm mentioning, these sort of operational and financial, administrative, or even scientific issues. And that's why this is really in our purview to address. We pay very close attention to all of these factors to figure out how to address them. Because if we have 4 arrows going in, yet only 1 arrow coming out, that's not going to cut it. We want really to make sure that we're having 4 arrows going in, and there's no slowdown or other factors in this pipe, so that we can have all 4 arrows coming out. We want to go full bore as much as possible. So the other thing I just want to mention is that the more we understand this process, the easier it is to get from point A to point B, as I showed you in that previous slide. And the more predictive it becomes if we can understand it better. But it's more complex than that. And so it's not that easy. When you look under the hood of this chevron diagram, for example, we need to really detail what is really involved at each process of this Chevron diagram. So we've mapped it out so that we can get more broad and more specific activities at each step of the pipeline. This is called the 4DM, the Drug Discovery, Development, and Deployment Map, and it's one of the widely used tools by researchers within the community to really understand the steps along the translational pipeline. Each section here is color-coded so that you know where you are on the map, and it's linked to this other Chevron diagram on the left. And this allows you to find information and best practices or to connect with relevant programs at NCATs or other types of resources. So although it's daunting, although it looks very busy here, it is not insurmountable. And so when you know where you're going and you know where you are, then you know what you need to do for your next steps. You know you might need to identify collaborators or other experts that you need to engage. And so you can find the expertise to help. This map was developed by members of the Action Collaborative of the Forum on Drug Discovery, Development, and Translation of the National Academies of Sciences, Engineering, and Medicine. And it has been an incredibly useful tool. So I encourage you to take a look at this. It is on our NCATS website. So essentially, this is really what the NCATS Way is. We are trying to use these tools and resources, but also really outlining what translational science is so that we can use this approach to hack into the science and advance it. And the ways that we do that are we bring together stakeholders that haven't talked to each other before, and we include patients. We need to have these cross-cutting collaborations and partnerships. We need to find and develop sort of these robust platforms and technologies and engage transformative stakeholders into the discussions as we develop those. And then we need to basically have a very deliberative approach in how we think about risk-taking. We need to have a very productive risk-taking to remove those scientific and operational barriers as we move forward. So the NCATS Way, it allows us to start to chip away at the big operational and scientific challenges that tend to build up in that pipeline that I mentioned before. So some of the operational roadblocks we encounter are in the areas of education and training, part of an effort that we need to fill the gaps for getting people more involved in translational science and bringing their learnings to their own backgrounds and what they're doing. Or, for example, data transparency and IP management, which are not very compatible. But when you have to be involved in an environment of team science, which is what NCATS is really supporting for the most part, team science is really critical to what NCATS is doing. And so data sharing must be unabated for projects to prosper. So how do we ensure that it's done in the way that protects intellectual property or IP? And of course, the other side of the coin on the scientific problems, these have been somewhat intractable over time. Things like predicting human toxicity and efficacy from the preclinical perspective, and encouraging and establishing better data interoperability and strengthening our clinical research and clinical trials infrastructure so that we can get more treatments to more people more quickly. And that, of course, is our goal. So, I mentioned that that our mission really is not disease focused. It's really about all diseases. So because it's about all diseases, that means we can't go one disease at a time. So we use two main translational sciences approaches to think about how to address rare diseases and other diseases in a more than one disease at a time way. So on the left-hand side, everything that we do is about a collaboration. We say that translational science is a team sport. We have the expertise and the technologic capacity to cover the breadth and the preclinical and in the clinical space. And we rely on our partners to cover the depth of a given disease or needed project. And the second thing on the right-hand side is the development of platforms and tools and resources to support the breadth of that activity. And both of these strategies help us save time, lower costs, and get more treatments to more people faster. So I hope you're really sensing a theme here. So NCATS oversees the largest research portfolio on rare diseases at the NIH, and it's led by Dr. Ann Pariser. Most of what I'll present today going forward is work done by Ann and her group, along with a long list of collaborators across the NIH, other agencies, as well as the private sector. So I'm going to touch on essentially what the Office of Rare Disease Research does. And it's essentially, we can break it down into three basic areas. The first is knowledge and data. And so we have a variety of programs that we have in this space that I'll talk about in a moment. We have research and collaborations that another series of programs in this space that I'll talk a little bit about. And then community engagement. And I'm going to touch again on all these aspects going forward, but I wanted to give you a sense of sort of the breadth and the depth that the Office of Rare Disease Research does at the NCATs. So moving forward here, I want to give you a sense of, and I'll focus a little bit more later in the talk on two specific projects that are using a platform-based approach. So I want to begin by just talking a little bit about some of the NCATS resources for patients and patient advocacy groups. First of all, we have something called GARD, which is the Genetic and Rare Diseases Information Center. This is a resource for patients and patient advocates and caregivers and clinicians alike. A variety of information about specific rare diseases. You can find out information on clinical trials, treatments available for specific rare diseases, and even what the different rare diseases are in lay language so that it's accessible to the community. We also have something called the Toolkit for Patient-Focused Therapy and Development. I think this might be a very good toolkit for this community to make sure that they are aware of and know about. It identifies things like how to understand the translational research tools that are available, how to determine patient needs, how to facilitate scientific collaborations, and how to even fund discovery research. So there's a lot of information in this toolkit that's available for patients and patient communities to look at. So I encourage you to do so. And then the last one here that I'll mention is RADAR. This is Rare Disease Registry Program. And here we work with researchers to pick an existing data standard, if possible, if there are data standards out there that fits the research questions and that they can be both validated and interoperable. So it's a patient registry that you can identify, perhaps become more involved in, and use it as a way to access other types of information and define ways in which you want to harmonize data to move forward. So I think this is an important project as well that NCATS has. And so I encourage you to take a look at all of these different programs on the NCATS website. Now I want to take just a brief diversion here because this outlines one of the main problems within the rare disease space and electronic health records space specifically. And the COVID pandemic has really heightened this awareness, and it's something that I wanted to just mention here. Another program that NCATS has been doing is to think about how to bring in electronic health record data related to COVID to help make sure that we can speed research and improve patient care. And to that end, we needed to develop a secure national resource of electronic health records to do so. So for the most part, we partnered with the centers, the Clinical and Translational Science Award programs, the Clinical and Translational Research Award programs within the IDEA states through NIGMS, and then the Center for Data to Health, a part of the CTSA program. And what we've done is we've highlighted here the idea of how to harmonize data from electronic health records to make sure that they can be available for researchers to use. But the big problem has been this idea of interoperability. But I'll tell you in a minute how we've solved that problem. Right now, we have over 2 million COVID positive patients in this database, and we have over 6 million total patients who are uninfected as of within this database as of today. And so there's a lot of information in here, and we're hoping that people who can access this data will be able to make these sort of informed decisions as we go forward, especially as the pandemic unfolds and we start to enter more of a chronic phase, and how this virus is going to be impacting lives in the longer term. On this dashboard that I'm showing here on this link, you can find vaccine and medication data. You can also see the data contributors as well as comorbidity data. So I encourage you to take a look at that. But the important piece of information here that I just wanted to relay is this idea that the data are in electronic health records are very disparate, and each institution across the country who has a healthcare organization generally has a way in which they prefer to use their common data models for that type of information. But they're very different. So for example, one institution might collect height in centimeters. Another institution might collect height in inches. And so what we're trying to do is make sure that when we pull these data together, that we're actually mapping them onto just one of those. We're going to pick one and map everything to that so it's all consistent and we're comparing apples to apples. So this is incredibly important. But the problem within rare diseases is that, generally speaking, the diagnostic codes are lacking. And we just don't have the detailed information needed to make sure that we're able to see rare disease patients in these kinds of databases. And so that's something that I think we need to do a little bit more in moving this forward. And I'm hoping that this is one thing that might help. This is, one of the obstacles to developing rare disease treatments is to include the patient diagnoses, and there are small numbers, and rare disease patients are geographically dispersed. There are fewer people who are working on these sorts of diseases. There's a lack of natural history studies. There's missing biomarker data. So there are lots of challenges for this particular approach, but we're seeking to tackle these challenges by working to identify the common problems among rare diseases and addressing them collectively rather than individually. And that's exactly what the Rare Disease Clinical Research Network is doing, the RDCRN. So this is an NCATS-led program that partners with 9 institutes and centers at the NIH, and it really aims to address many of the unique challenges that researchers face in developing rare disease therapies. Currently, this network consists of 20 individual consortia with representatives in different patient advocacy groups and rare disease communities. And as we become more sophisticated in our ability to manage access and use healthcare data, as highlighted from that N3C resource that I just mentioned, I expect this is going to be a group that's going to be able to tackle some of these issues. But certainly with rare diseases, the issue is magnified because our healthcare system is based on billing, and billing is based on diagnoses. So this is another big health inequity that we need to really address for the rare disease community. And I look forward to doing this, tackling this problem as we continue to move forward. So I also wanted to highlight some approaches we've used at NCATS to help draw awareness to rare diseases and to engage with the community with these approaches. On the left, we held a challenge competition for the last 2 years to raise awareness through our Rare Diseases Are Not Rare challenge. And if you haven't had a chance to watch this video by Jacob Thompson called Keep on Fighting, I encourage you to do so. It's filled with inspiration and hope. In addition, we have a variety of workshops every year, and here's the latest one on the right that I'm showing. And this is called Gene Targeting Therapies, Early Diagnosis and Equitable Delivery. And importantly, as I mentioned before, we invite patients and patient advocacy groups to provide comments at these meetings because it's very important that we hear their voices and hear what they're really thinking about and understanding how we can move forward better together. So this is another area that we are promoting. And the slide's very busy. I don't need to really go through much of it, just to say that it is true that patient memberships on these sorts of ideas and workshops is actually disruptive technology for rare disease therapeutic development. And so that's why it is very important that we have patients and patient advocates at the table. So now I'm going to talk a little bit about a study that we've affectionately called Novel Explorations on Rare Diseases, but that acronym turns into NERD, and so we've changed the acronym to say ideas. But essentially, the idea here is that we want to really get at this idea that data is limiting here in rare disease research. We know that there are 77,000 rare diseases, but we have no firm number. We think that it's 30 million Americans who are affected with rare diseases, but no firm number. And there's a several-year delay in accurate diagnoses, the diagnostic odyssey. And this is a big problem in rare diseases, as you know. And the direct and indirect costs have been unquantified. And as I said, ICD codes don't exist for most rare diseases. And so this leads to poor tracking and our inability to really see what's going on in these diseases. So because of this, sorry, one more back. We want to get a better understanding of how the disease burden is on rare disease patients, and this is going to inform the research priorities for rare diseases. And so that's exactly what we're trying to do here. We have established 5 partners for this program. Along with NCATS, we have a couple of academic medical centers and a health insurer and an industry data analytics company. And our goals have been to establish methods for analyzing EHR data in the rare disease community, to help develop algorithms that are needed to help make those more robust and actually see them in the database, to map selected patients and their disease journeys, to quantify and describe really what that medical utilization looks like. And then to identify features that may signal a patient at high risk for having a rare disease before they're even diagnosed or before they see their 10th doctor, for example. We want to get at them earlier so that they can have a better chance at diagnosis. So we want to help characterize what we're looking for in these databases so that we can understand a little bit more deeply what's happening before diagnosis as well as after diagnosis, so that we can help inform our ways in which to identify rare diseases out there that need the help. We'll also be able to quantify direct medical costs per patient per year as age-matched controls as well. And so this is part of the idea behind this particular approach. And it also is important in the sort of following on the heels of the Every Life Foundation report that you've probably heard about, where they provided a staggering number of the non-medical as well as medical costs to patients and families at approaching $1 trillion for those families. So this for those costs. And so this is really a big problem within the rare disease community that we need to tackle. So our pilot study on this approach is to take 14 different rare diseases and to look at sort of the diagnostic odyssey in the electronic health records that we can see from our partners. And we were looking for rare diseases that we did know have ICD codes so that we could pull data that we could know about, and then we'll be able to build that out more for rare diseases that may not have ICD codes. And so this is also some of the work that has, this is overlapping, but a little bit distinct from the EverLife study. And I think it'll really add value to the discourse as we start to raise more awareness about these needs in the rare community. So this approach, where we've just submitted our publication, and so it's been sent to the journal. We haven't heard back yet, but essentially, we hope that this will identify rare disease patients in healthcare, make them more visible in the healthcare community, understand the diagnostic odyssey. It's real and it's costly, and also identify costs of care for rare disease patients, and understand that compared to the age-matched controls without rare diseases. So we think this will be an important study, and stay tuned for when that comes out. We're very excited about that. So I'm going to move on to some treatment approaches that get at sort of therapeutics in this space, and touching upon sort of the more than one disease at a time approach. And this is work done by the Office of Rare Diseases and Parasur, as well as the NCATS labs through Don Lowe and many of his colleagues as well, among with, again, many other collaborators as well. What I'm showing here is how we're thinking about the development for more than one disease at a time. And the important thing too here is that these approaches, we're specifically looking at rare diseases, but we think they will have a pretty big impact on common diseases as well. So I want to start by just talking about this approach and devising this sort of platform-based approach. And since we know that 80% of rare diseases are caused by a single gene mutation, then genetic mutations then become the common thread of what we're trying to get at. And so we can create platforms that then will tackle that common thread. So we're looking at gene therapy, which is replacing a disease-causing gene with a functional one. And so we have a pilot for gene therapies. We're looking at one particular vector called adeno-associated virus 9. And we're trying to make this particular vector through a single manufacturer to address 4 different rare diseases. And we hope that we'll be able to identify IND-enabling work that can generate templates for other researchers to use moving forward. And in that way, we hope that it will speed research for many diseases. Another approach is looking at gene editing. This is essentially changing out a piece of DNA that's causing a disease. And here we want to expand the number of tools and delivery systems and assays and for safety and efficacy, as well as disseminating the resources as well. And then finally, another approach that we're using is something called antisense oligonucleotides, which is basically these bind to mRNA in cells that are producing disease-causing proteins. And what we're able to do here is look at one particular gene that may be responsible for a variety of different diseases. And that's the kinesin heavy chain isoform 5A, shown here, can get at Charcot-Marie tooth, hereditary spastic paraplegia, ALS, and frontotemporal dementia are some of these diseases that we hope that this will address. So these are a variety of approaches that we're piloting now to see if we can really look at this and get at these platforms for more than one disease at a time. But I'm going to just give you a sense of really how this works in a program called Paving the Way for Gene Therapy. And I'll just go through the fact again that we're looking at developing one AAV vector, or this adeno-associated vector, looking at the same sort of way of administration, and then looking at how we can produce this on a larger scale and have this purified in ways that will make this process sort of much more reproducible and predictive. And then we're going to make all of these data available for the research community. So what this looks like essentially is shown here. This is sort of a rendition of what this AAV looks like, but I'll just make it more cartoony here. Essentially, it's got DNA inside of this capsid. And what happens then is what we're doing for gene therapy is we take the viral DNA out, and then we replace that viral DNA with human DNA with a good copy of the gene that happens to be mutated in the specific disease that we're looking at. So we can look at that these different types of DNAs, add them into the vector, and then move forward to putting these into patients and hopefully fixing that mutated version of the disease. So this has a lot of hope underneath this particular approach. So we're going to be developing gene therapies for 4 diseases, 2 organic acidemias caused by these 2 disease genes, and then 2 congenital myasthenic syndromes caused by these 2 disease genes. There are a variety of reasons we picked these particular diseases largely because of the size and our familiarity with them. But then we're going to do this IND enabling work to scale it up, to do GMP manufacturing, and to make this available for then putting into clinical trials with our partners at the clinical center, as well as NHGRI and NINDS, who will move this forward and hopefully make this a reality. So we're really hoping that this is going to be a pilot program that works and that we can put these data into the public domain. We're currently at this stage here still working on the IND enabling work and the models that we've developed to make sure that everything's moving forward. So we have a little bit more time to go, but this is the idea behind this type of approach. And, I think this gets us back to where we started then. Our hope is to get more treatments to more people more quickly. And on the right-hand side, this is very aspirational, but it's not out of the realm of possibilities to think about how we can then move it forward to increase the treatments for diseases in the next 15 years. So I don't know where this purple bar will be at that time, but I'm hoping that it will be certainly higher than where it is now. That's our hope. And I just want to end on 2 more points. One is that if you're interested in looking at other kinds of funding opportunities available within NCATs that aren't typically around the realm of science, this one is in ethical issues and translational science. And I just wanted to point out that we are looking at ethical implications of development of these types of platform-based approaches to make sure that we consider these types of issues as we go forward. And then lastly, I'm just going to end on this slide. I want to mention that the thinking about health inequities is such an important piece of the rare disease community. There are many efforts in diversity, equity, and inclusion that are happening now, and not only from the standpoint of Global Genes with their Rare Health Equity Leadership Council, but also within the NIH. We have something called the UNITE program, and each of the letters of UNITE stands for a particular subgroup of individuals that are working on areas to think about how we can bring the idea of structural racism into the forefront of how we're handling research into the community for all communities. But we know that these are even more hampered and emphasized in the rare disease community. So I think that we're going to learn a lot from all of these types of approaches and convenings as they go forward. And I look forward to thinking about continuing to implement these moving forward in the ways that we're approaching it. So our ultimate goal is to get more treatments for all people more quickly. And I will end there and thank you so much for your attention. And I'm happy to open it up and turn it over to Donna.
Dr. Redder, thank you so much for that remarkable talk. You have lit up the question section. So I will try to get through as many as we can in the next 10 minutes or so. I think there are a cluster of questions which really are around the thoughts of how can patients truly drive the research agenda. I love the idea of patients, of us as disruptive technology. So how can we continue that momentum, that cultural change, that paradigm shift of patients, not only being included in research, but also really driving that change? What can patient advocates and patient advocacy organizations do? What can the researchers do and government agencies? How can we make that accelerate?
It's a fantastic question, and I might have to. It's a big question, so I want to kind of carefully walk through some of these ideas, and then maybe we can also unpack some of them as we go forward. But it's such an important question because, as I said, patients are disruptive to the process, and so how do we make sure to include them? And I think there are a couple of key points here. I think first and foremost, patients and patient advocacy groups have to be part of the discussion, and I think that for us, this is what we built into how we do our workshops and our meetings that we have, our convenings, to include patients within that discussion. However, we may not be able to reach everyone, and so I think if there are ways in which we might be able to raise awareness of the particular workshops, convenings that are happening to a rare disease community, a rare disease leaderboard, for example, to help people understand what might be going on so that they can tune in and be at least a part of the discussion, to ask questions or to raise points and to comment. So that could be one thing. There are a variety of other things too, things like meeting patients where they are. There are many other platforms that we need to consider. Not all patients have access to broadband internet, and or have disabilities that prevent them from being able to use particular devices. And so how we can make sure that we include that in our thinking of sort of the diagnostics that get developed, for example, and how those diagnostics can be used. I think that's another piece of the conversation. But when I talk about also meeting people where they are, I think other platforms like social media and groups on social media could also be very helpful in engaging in the conversation and continuing to raise awareness for things that they have. I'll also just say a few more things. One is thinking about how we can raise awareness by including people in the workforce who have rare diseases or are from diverse communities and communities of color. Researchers and practitioners and community engagement folks alike, and really making sure that we are using ambassadors, if you will, to engage the community as well and to reflect the community that they're representing. That's going to help us build better tools and better resources that are then accessible. For example, if we're working in a primarily Spanish-speaking community, we want to make sure that we have our materials in a language that they can understand. So creating materials in the Spanish language will be very important. So I think those are the kinds of things that we can do to really make sure that we're meeting people where they are. So those are just a few points that I would mention.
I think that's fantastic. And so I'm going to say to the group, I will not forget the ICD-10 question. We will get to that before we end. But I wanted to pick up on that last point about those very tactical points of engagement that you were making to ensure health equity and diversity in this work. You know, I am honored to sit on the Global Genes Rare Disease Diversity Leadership Council. And you know, we thought through a lot of these issues, and certainly, you know, one of my mentors, who's a former FDA administrator, Rob Califf, always says that incentives matter. And I noticed when you describe the NCATS Way, and you're thinking that aligning these incentives around different changes that you wanted to see, how important and what incentives do you think moral, ethical, or pragmatic will help make a real and long-lasting shift in the structural racism in research, but also in moving these very tangible, concrete efforts, as you mentioned, in doing in language, in culture outreach, but also in ensuring that the gaps in data include the fact that there are people of color who do have rare diseases. So how does that work? How do you incentivize that work?
Yeah, that's a great question. I think that there are a variety of ways in which we can do this. And it really, I don't know if this is a truly satisfactory answer, but I think it really goes back to this idea of education and training and awareness, and how we approach the communities to incentivize the communities to get involved. And in a way, it then becomes part of an ecosystem. And so then when the communities are involved, it forces the scientific community to understand and pay attention to what the needs are of the communities. So for example, just building diagnostic tools, again, thinking about building those tools in a way that then, if you build it as a particular as a particular app that requires access to an internet, or if it's an app that requires dexterity that may not be accessible to everyone, then we're not really addressing the deeper problems. And so that engagement, that awareness and understanding to the scientific community of what the needs are for the patient community is really important. And then vice versa, the understanding to the patient community of what kinds of things are out there that are being considered, so that we can hear those voices and include them in how we're approaching the next steps. And I'll say, I can't tell you how many patient communities I have heard from who sort of think about their own genetic disease and you know, over the course of the pandemic, how it might have been deprioritized or sidelined due to the focus on COVID, let alone worrying about COVID itself. And so the work that we're doing in sort of these diagnostics and platform approaches to getting at these therapies, and talking about them in ways that the patient and patient advocacy groups can understand, so that they can help us move that information and the science forward, I think would be really just a huge value. So awareness, education, training, understanding that sense of urgency, and working together. I think those are some of those incentives, but nothing speaks louder than providing the resources that are needed to enable the work.
Fantastic. So really quickly before we end, because I don't want to get in trouble with this audience who's dying to know about how that their rare disease has an ICD-10 code, to go about the process of getting a code if they don't have it, and being a part of that whole project. And I'll just say, from the Global Liver Institute's perspective, we have engaged in that work and getting additional coding in non-alcoholic steatohepatitis in NASH. And so tell or at least direct folks to where they can get more information on how they can help push with ICD-10 coding for their disease.
It's a great question. Actually, Donna, I'd love to hear more about how you did that within the liver community. That sounds like the ideal way to go. So maybe I can turn it back over to you. But, you know, I do think that this is really important because, you know, a lot of the rare diseases are invisible in the systems until we figure that out. some of them do have ICD codes, but they're not quite right. They're codes related to developmental delay, for example, or limited growth, or these very sort of basket terms in the ICD codes that then have been sort of used and as a proxy for rare diseases, and we do need to do better. I think part of it is really an understanding of what those look like. And so again, I'm going to turn over to you, Donna, to see what your process was, because that sounds like a really good one to follow.
Okay, well, we'll work on that. Thank you so much, Dr. Redder, for this conversation. Clearly just the start.
Absolutely, absolutely.
So for everyone, we appreciate your feedback in this session. Before you go on to the next session, we're going to need you to respond to the polls that are being launched. We want your feedback so that we can continue to provide great content and programming like this, clearly. You can also share your feedback via events@globalgenes.org. Up next, as if that wasn't enough. I mean, this makes my morning. But up next, please head over to one of the breakout sessions on either basket and umbrella trials or advances in clinical trial design and real-world evidence. Additionally, I want to remind you that we will still be hosting office hours and informal networking rooms after the official close of the program at 3:30 p.m. And where are you going to go on a rainy day and everything like that? Stay for the networking. Please join us to discuss the content of the day, meet your peers, connect with speakers and experts, and we want you to walk away with a robust toolbox to advance research in your own community. Thank you again for joining this keynote session. Enjoy the rest of your day.